Hi everyone, today in this video I am going to show you how to properly use the centrifuge in routine lab work. So here we are using the benchtop centrifuge with a fixed angle rotor. So first what you're going to do is check whether the lids of the tubes are properly closed. Then we need to group the tubes in pairs according to the volume of blood. It might not be the exactly same, but we can choose approximately equal volumes together. Now we take the pairs one by one and put the two tubes inside the wells as we show here in the opposite direction of the rotor. This is called the balancing of the centrifuge. Then we close the lid of the centrifuge and set the RPM to 3000 because we are going to separate the serum here, and for that we need to centrifuge the red top tubes in 3000 to 5000 RPM for 10 to 15 minutes. Tube placement doesn't always have to be in a cross pattern. You can place them in any position as long as the centrifuge is balanced. And also, each time we do the centrifugation, we need to check the RPM and the time and then start. Always make sure the tubes with equal volumes are placed opposite each other because the proper balancing is the key to safe centrifugation. If the centrifuge is not balanced, that will make very harsh sound and it will start to shake. And if the centrifuge making the sounds and shaking, we need to power off as fast as possible, and then before opening the lid we need to check whether the rotor is completely stopped, and then we can open the lid. To stop the centrifugation we can do manually with the power off button, and also we can put the time limit and it goes off by itself. I hope you gained some useful knowledge about correct centrifugation techniques from today's video. So always remember, correct balancing protects your samples and your centrifuge. Thanks for watching LabVerse. Learn smart, work safe in the lab. See you in the next video. Bye bye.